Okay, everybody, this is the second part of the glycolysis lecture. I have already explained the process of glycolysis right here. You should be aware of it and you should memorize it. Now we're going to go to the important part, which is glycolysis regulation. Actually, this is more important than the process of glycolysis itself, because the teacher herself emphasized this part very, very much. And I want to give you a disclaimer that everything I talk about and all the details are in the description of this PowerPoint. So to start, I have put the process of glycolysis here in short, and this information you should know. Now, what is the fate of pyruvate? If I go back to glycolysis, we start with glucose, and then all of this we end with pyruvate. And it's asking us, what is its fate? Let's say we are in a normal cell. We know that glycolysis doesn't need oxygen to work, right? But if the cell is aerobic, right, it needs oxygen to breathe or to have cellular respiration. It means that after glycolysis, pyruvate is going to enter into the citric acid cycle, which needs oxygen. And then after the citric acid cycle, there's in the mitochondria, there's the oxidative phosphorylation, or it's called the electron transport system, which also needs oxygen. So, pyruvate, if it's in an aerobic cell, or if the oxygen is high in the cell, it's going to enter the citric acid cycle, and then in the mitochondria, it's going to enter the electron transport system. So, if pyruvate is in an anaerobic cell, or a cell that has a low oxygen supply, then that means pyruvate is going to be converted to lactate by this enzyme called lactate dehydrogenase. Now, what is this reaction? So basically, this is pyruvate and this is lactate. By the enzyme lactate dehydrogenase, it becomes lactate. Where does this happen? In cells with low oxygen, such as skeletal muscle cells that is exercising and red blood cells. If it doesn't have the mitochondria, then it won't have the electron transport system that needs oxygen to generate ATP. So it depends, in the red blood cell, it depends on glycolysis for ATP. Okay? Now, lactate dehydrogenase, as I said, is the enzyme that catalyzes this reaction between pyruvate and lactate in anaerobic cells. Okay? So, these, she didn't really stress on them, but you have to know that lactate dehydrogenase has 5 types, and each type is found in a specific tissue. There are 4 polypeptide chains and so on. And if you're interested to know more about lactate dehydrogenase, I had already explained it to Year 1 students, and you can click on this link right here and you can watch the part of that video that I explained. So basically, this is the reaction. Pyruvate plus NADH gives us lactate and NAD+. So let's take 2 examples of LDH1 and LDH4. LDH1 is found in the cardiac muscle. LDH4 is found in the skeletal muscle. In the cardiac muscle, we want pyruvate, not lactate. So the reverse reaction is going to be longer than the forward reaction, because all of the lactate are going to become pyruvate. We don't want lactate in the hearts, right? Because if you recall, when you go to the gym and you come back home and you feel the soreness in your muscles, that's because all of the pyruvate is becoming lactate. And when lactate is accumulated in our muscles, it causes this type of pain that you feel, okay? Normally, lactate is removed by the kidneys, but when it's accumulated, not all of it is removed, and it causes the pain that you see. So, of course, in the heart, you don't want to have lactate. You want pyruvate. So all of the lactate is gonna form pyruvate. That's the case of the cardiac muscle. Low Km and high Vm. What does that mean? Low Km means the enzyme lactate dehydrogenase has high affinity to lactate. It means that even at low concentrations of lactate, the enzyme binds very rapidly to lactate and converts it to pyruvate very quickly. Okay, that's exactly what low Km and high Vm means. Now, if we want to talk about a skeletal muscle, of course, as I have just told you that you're gonna have an accumulation of lactate in the muscles after you've exercised. So, of course, the forward reaction is going to be favored over the reverse reaction, and we're going to have more lactate than pyruvate formed in the skeletal muscles. So, low Km and high Vm. Low Km meaning high affinity to pyruvate, and high Vm, it means it converts it very fast or very rapidly to lactate. Now, if you also want more information about what Km and Vm is, I also prepared a video for the year 1 students. It's more detailed if you're interested, but if you're not, I think it's enough for you to know that low Km means high affinity and high Vm means there's a fast or rapid conversion. Now, this is important. So, the free energy changes in glycolytic reactions. So, as you can see, these are the 10 steps of the glycolysis pathway. And this table is trying to tell you that there are 3 reactions in glycolysis that occur spontaneously. So, spontaneously means that the delta G, or the change in the free energy, is negative. That delta G is negative, this means that the reaction occurs spontaneously. It means that it does not need a lot of energy for it to occur, okay? It means that this reaction is energetically favorable. Now, which reactions are those that are spontaneous? One, of course, they're the ones in red. If I can help you to memorize these steps, I would say that the first step, which is the step where hexokinase, the enzyme, takes glucose and locks it in the cell as glucose 6-phosphate. You can memorize it in the form of that the first step is the step that locks glucose into the cell. Okay, that's the first spontaneous reaction with delta G negative. The second step is the step that prepares the long chain of fructose 6-phosphate to be broken down into DHAP and G3P. So, the first spontaneous reaction is the one that keeps glucose imprisoned. The second step is the one that prepares the chain for it to be broken. Okay, this is the first one. The second one. The third one is the product that we need. Okay, so we're working all this way to reach the product that we need. So what are the 3 reactions? The first spontaneous reaction is locking it in. The second reaction is preparing it for breaking it down to 2. And then the last one is our product. Okay, now, as I said, regulation is the part that she cares about the most. So how would you regulate glycolysis? You would regulate glycolysis by controlling the receptors, which are called GLUTs, and by the enzyme that is called glucokinase, or if you know it, it's called hexokinase, right? So GLUT has types like GLUT2, GLUT4, and whatever. And glucokinase also has types. It's also called hexokinase in, actually it's called glucokinase in the liver, and it's called hexokinase elsewhere. Now, if I want to give you an example on how it's regulated, let's say that you have high glucose in the blood. So what happens is that this is the cell, and you have a receptor GLUT2 here in the liver, for example. And what happens is that it's going to take glucose in very fastly and very rapidly. That means it has high Vmax, right? As I said before. So GLUT2's high Vmax ensures efficient glucose transport into the liver and pancreatic cells when glucose levels are high. Check. So moving on to glucokinases, as I said, the receptor did its job. Now it's time for glycolysis, or the first step of glycolysis, which involves glucokinases or hexokinases. It has high Vmax and high Km, which means that it's working fast in locking the glucose into the cell, but it has high Km, which means that it doesn't have that much high affinity to glucose because it's working efficiently in phosphorylating glucose, okay? It needs to work efficiently, or more precisely, in phosphorylating glucose and to keep it in the cell, okay? Even at moderate concentrations, it allows the liver and pancreas to sense and respond to changes in blood glucose levels effectively. Now, so basically, as I have just said, you have a high insulin level in the blood. So let's say you are in the pancreatic beta cells of the pancreas. So glucose binds to the GLUT receptor, and it comes in and performs glycolysis, right? This is normal, we know this. And then after glycolysis is done, we have ATP, right? So what ATP does is that it goes to the potassium channels and it closes them. So when it's closed, the voltage is increased in the cell, and this causes depolarization. Depolarization then causes the opening of the calcium channels, and this leads to an influx of calcium into the cell, and this calcium stimulates the insulin granules to release insulin into the circulation. So insulin then goes where? It goes to the liver, and it can go to adipocytes and to myocytes. Okay, so what insulin does in the liver is that it stimulates glucokinase. As I said before, glucokinase is the same enzyme as hexokinase, you can see right here, but it's called glucokinase in the liver and hexokinase in other cells. Okay, so insulin stimulates glucokinase. And when glucokinase is stimulated, what that means is that we're stimulating what? Glycolysis. Okay? So this increases, glucose uptake is increased, and we increase the utilization or the usage of glucose in the form of glycolysis. Okay, now moving on. How does insulin cause the increase in expression of these GLUT receptors? So basically insulin binds to its receptor, right? And then this induces the GLUT transporters that are found in vesicles to fuse with the plasma membrane, and when they fuse with the plasma membranes, they respond to glucose and they bind to it, and glucose comes in and we have glycolysis, right? But then when insulin levels are lower, okay, when it has responded and insulin levels are lower, these GLUT transporters fuse again into the vesicles and come back into the cell because they're not needed right now. Okay, so what does she want us to learn? What is the effect of insulin on adipocytes, myocytes, and hepatocytes? This is an easy question. So basically, insulin, what does it do in these three cells? It upregulates the expression of what transporter? GLUT2 or 4, depends on what cell you are talking about. So insulin upregulates or increases the expression, okay, of glucose transporters on the cell surface. So here I wanted to show you that the product of this reaction, which is glucose and hexokinase, can actually inhibit hexokinase. So hexokinase's job is to keep glucose in or imprison it, right? So when it sees that there's high levels of glucose 6-phosphate that is imprisoned, it itself can inhibit it to stop imprisoning more glucose because this is enough. Now, so if you just look at this graph and you see that there is enzyme activity and glucose concentration, you can compare these two enzymes at low levels of glucose. For example, you can see that, for example, this is 2. Let's say it's 2. You can see that this enzyme's activity is much higher than this enzyme's activity at 2 millimoles of glucose. This tells us that hexokinase, which is the enzyme that's related to the muscle, has higher activity than hexokinase that is in the liver at low glucose concentrations, right? But as you increase the glucose concentrations, if you see right here, this enzyme goes into a plateau, if you can see right here, but this enzyme continues to increase, right? It continues to increase. So this tells us that hexokinase, which is the liver enzyme, okay, has higher activity than hexokinase 1, okay, which is related to the muscle, at higher glucose concentration. So I will reiterate, this one, or hexokinase 1, has higher activity at low concentration, but as the concentration gets higher, this one is going to have higher activity than hexokinase 1. This also tells us that hexokinase 1 has a maximum activity it can work in, or hexokinase 1 operates near its maximum capacity and cannot respond to further increases in glucose concentration. So what does she want us to know? What enzyme do you expect to work faster in response to lesser glucose and to higher glucose? So lesser is this one, higher is this one. Now, this is important. This is very important. Actually, we had a question in our exam last year from this. So you have glycolytic flux, which is the rate of glycolysis, and the enzymes. So basically, this graph is telling us the effect of these enzymes on the rate or the speed of glycolysis. As you can see, this one has a higher, it increases the rate of glycolysis more than this guy right here. So she can ask you, or her purpose of this graph is to ask you, what is the rate limiting step of glycolysis? You cannot say it's hexokinase, because if you want to define what a rate limiting step is, it is the slowest step in the reaction of glycolysis, and the speed of this step actually affects the whole rate of glycolysis. So the slower this reaction, the slower the whole glycolytic reaction. The faster this one, the faster the whole reaction. So if she asks you in the exam, what is the rate limiting step of glycolysis, you're gonna say phosphofructokinase 1, PFK1, not hexokinase, because this one is fast. Okay? Moving on. Professor actually didn't have any details about this in the lecture, but I'm gonna be so general about this because I didn't find any details about this, and she hasn't asked about this in my information, so I'm gonna be very generalized about it, and I'll get into it. So basically, this on the left side is glycolysis. As you can see, we have pyruvate at last, but this is called the Rappaport-Lou-Behring cycle, which is a cycle that happens in the red blood cells. Okay, so what happens is that in glycolysis, 1,3-bisphosphoglycerate is going to become 3-phosphoglycerate and then pyruvate, right? But in this cycle, this 1,3-bisphosphoglycerate, instead of becoming 3, it becomes 2,3. And you're going to ask me, why is that? Because in RBCs, we have hemoglobin, right? And hemoglobin is bound to oxygen. And when they reach the tissue, hemoglobin needs to unbind to oxygen so that oxygen can leave and enter the tissue. So the thing that helps oxygen unbind from hemoglobin is 2,3-bisphosphoglycerate. So it binds, and then hemoglobin lets go of oxygen, and then oxygen can go into the tissue. That's it. In essence, that's what this cycle is. Of course, I'm going to say it again. Please read the notes that I have written in the description, and this is the end of the regulation of glycolysis.